DNA Testing Center as a standalone business from the Department of Applied Biology and Biochemistry. That's where we originated from, in the Faculty of Applied Sciences. So the Agtc, as we call it, was established to offer services in this biotechnology field of DNA analysis. And we are running it as a business. All the services that we offer here are paid for. And we have clients who come in for their paternity test, they pay. And even government departments, the police, the forensic departments of police, and even the civil protection unit, they use our services a lot and they pay. And we envisage that in the next 5 years we should be weaned completely from the university and be a standalone entity that is wholly owned by the university, but no longer running under the direct management of the university. To work in a setting like ours, in a DNA testing center, we recruit mainly people who have degrees in applied biology, biochemistry, mainly specializing in molecular genetics.
My name is Mlilwana, and my journey to wearing the white lab coat began here in Zimbabwe at the National University of Science and Technology, where I studied for an undergrad in applied biology and biochemistry. I then ventured into biotechnology by studying at the National University of Science and Technology here in Zimbabwe, a master's in applied microbiology and biotechnology. I have since developed an interest in forensic genetics since I started working here at the Applied Genetic Testing Center in Zimbabwe, which offers various services for the nation, including Zimbabwe Republic Police and the general population. We were solving murders and rape cases. This then prompted me to then study a PhD in forensic genetics with the aim of solving all these cases.
My name is Agtc, a research fellow in the Department of Applied Genetic Testing Center. We do DNA analysis in this laboratory. So I'm going to show you the process that we take to analyze the bones. And this is a femur bone which have been collected from a dead person who is not known. So we need to identify this person. So to remove all the contaminants that might be on the bone, we have to wash it thoroughly. As you know, that DNA is very, very sensitive. So if I touch it without gloves, my DNA might be there, and the profile that will come there will be mine, not that individual. Or it will be a mixture of the DNA. So once I wash the DNA here, I have to dry it. Thoroughly, when it is washed, when it is clean, it has to be dried. We have to leave it overnight for it to dry. Once it is dried, then we have to grind it using the tissue which is there. I'm now switching on the tissue so that we can grind the bone which is inside the tissue. Now we are closing the lid so that we can run the tissue. Now it is grinding the bone which is inside the powder. Once it is in powdered form now, it is ready for DNA extraction. The powder now has been removed and transferred to the Eppendorf tube for DNA extraction. It's an automated robotic extraction machine called the QIAcube from QIAGEN. we put all the buffers in the machine and all the necessary accessories for extraction in the machine, and it will do all the work for us. And once we put everything there, then we close the machine and we start it for DNA extraction. Right? The automated machine, it takes 12 samples at a time. And for those samples, it takes about an hour for each to finish the extraction.
I'm just going to take you through the processes and steps that we go through after the sample has been extracted for a DNA sample. What we have here is a case of a missing person where the bones have been found buried, and a reference sample. A reference sample is a sample that is collected from a person's child who has been found to be missing, which is what we'll use to compare and identify the missing person. So what I have here now are different kits that we'll then use to amplify their DNA. When we have our DNA sample from sample extraction, we quantify it using the real-time PCR system to get a quantity of the DNA that we have in the sample. The DNA is then diluted to a standard concentration of 1 nanogram per microliter. We have various kits that we use depending on the type of test that is required by the client. And what this test does, it amplifies different regions of a person's DNA within the sample to generate many copies of it. Once that has been complete, we'll have a sample that is very little, about 10 microliters, and we use only 1 microliter of that. One microliter sample is used to amplify and electrophoresis, meaning to separate the DNA into different fragments using the state-of-the-art 3500 genetic analyzer. What this machine does is it separates your DNA into various fragments. So once we've done the PCR test, what we then do is we use the state-of-the-art 3500 genetic analyzer to electrophoresis and separate the various DNA fragments. We've loaded our plate into this cartridge, and we then load it into the 3500 genetic analyzer. Once this run is complete, we then analyze the raw data on our gene mapper software to generate our DNA profiles, which will look like this. So we have the various DNA positions that are shown here by the various colors, and the peaks that have been detected, which are the DNA that we are looking for. So once we've generated these DNA profiles, we are then able to compare a DNA profile from a bone sample and a DNA profile from a reference sample. And then we are then able to conclude whether this person has half of the reference sample's DNA and it's a complete match, and we're able to identify the missing person. So the cost from start to finish for each sample is quite affordable for our police officers and our general population who come and consult for DNA testing. Is to see the girl child being developed in science